Good morning, class. Hopefully you guys had a good weekend this past weekend. Let's go ahead and get started on today's topic, which we will be talking about tools tests for COVID. So as usual, the outline goes as today we're going to be talking about key diagnostic testing terms, which I'm going to be testing you guys on. So please understand them. If you have trouble understanding them, feel free to ask. And then the main lecture is going to be focused on covering the three main tools that we have to test against COVID. And these are reverse transcription, quantitative polymerase chain reaction, Anagen test, and the antibody test. And so again, like always, if you understand the learning objectives, you're going to do fine on the exam. So understand these 6 different terms that I'm going to go over. Really understand the sensitivity and specificity if I were to give you some info dealing with that, and know how these terms apply to the data given. Know what are the starting materials for each of these 3 tests, and understand how these 3 tests are detecting COVID, so the genetic material or the protein or the antibodies. And then just understand the basic science behind the testing. So let's start with the first term, sensitivity. So any good test is going to have high sensitivity and high specificity. With the term sensitivity, this is referring to the accuracy of the test that identifies the true positives, so the people that are truly infected. And so we'll use an antibody test example. So, if you have the antibodies present in your blood, then that means you've had previous COVID or ongoing infection, depending on, what antibody you have present. So in this example, the sensitivity of this particular kit is 95%. So this means that anytime that there's a blood sample that is being tested and has actually does have antibodies against the virus, it means that 95% of the time the test will register as positive. So this is going to be the true positives. Now, notice how it's not 100%. So there's always going to be some margin of error with some of these tests. And so what this means is that anytime that there's a blood sample that does have antibodies against the virus, meaning, there's infection present or previous infection was present, then this is going to register as negative 5% of the time. So that means 5% of the time that people who actually have been infected before are going to register as negative, and this is known as false negative. Now, with specificity, it's kind of looking at the opposite of this. So with sensitivity, it's looking at how accurate it is detecting at a person that's actually infected or has the antibodies present for the virus. With specificity, it's looking at the accuracy of the test that identifies true negatives or people that were never infected. So when we look at this specificity, it's for this particular kit, it's registered at 90%. So what this means is that anytime there's a blood sample that does not have antibody against COVID, it will test negative 90% of the time. So this is true negative. These are people that were truly never infected. Now, again, with specificity, there's always some kind of margin of error as well. And so what this means is that anytime there's a blood sample that does not have antibodies against COVID, meaning they're not infected, will register as positive 10% of the time. So this is meaning that this 10% are false positives. And so to kind of drive this point home, I think looking at pregnancy tests is, another way of trying to understand some of this terminology. I think it's a little bit more, abstract when you're looking at the COVID test, but with this, we have more of a concrete example because you can actually visualize some of this. So when we look at a pregnancy test, here we obviously have a woman that's pregnant and the pregnancy test comes back as positive. So this is a true positive, meaning the test is sensitive enough. Whereas this man obviously is not pregnant. And so this is defined as true negative. And this is going to associate with the term of being specific. Now, when tests are not sensitive or specific, then you're going to end up generating some false positives or false negatives. So in the context of a false positive, basically the test is going to register as positive, but indeed the person really is negative for the actual test of interest. So with this pregnancy test, this man obviously is not pregnant, but the test is coming back as positive. So this is how you refer to the term false positive. And this refers to this as not being specific. Now with this woman who, again, is obviously pregnant and the test comes back negative, this is known as the false negative. And this test is obviously not sensitive. And so when we apply this to COVID testing outcomes, we can, see the same thing where if a test comes back positive and the person's actually infected, then this test is sensitive and they're truly positive for the infection. Whereas if the test comes back positive, but the patient does not have coronavirus, then this is known as a false positive, and this is not specific. And here we have, the opposite now. A test comes back negative, but the person's actually infected. This, again, is known as a false negative, and the test is not sensitive. Whereas this patient is not infected and the test came back negative, and so this is true negative, or the test is specific. So sensitivity equals detecting true positives, and yes, exactly, perfect. Sensitivity, someone wrote in the comments, sensitivity equals detecting true positives and specificity equals detecting true negatives. Yes, if you can apply that concept to this next question, you will do fine on the exam, because I'm gonna basically give you guys a chance to practice with some of these questions. So on an exam, I might ask you, the diagnostic test has a sensitivity of X percentage, so here in this case, 98%, and a specificity of some percentage, in this case, 95. And what I want you to be able to do is identify these particular terms and apply this with this particular data. And so I've color-coded the terms with the respective term that I want you to apply it to. So what I'm going to do is just break you guys out into groups. So I want to make sure you guys are understanding these terms. For those that are struggling, hopefully your peers can help you understand this. And if not, we'll come back and I'll go ahead and walk you guys through the answers. So let me go ahead and break you guys out really quick. And then if you guys have any questions, feel free to call me. The people that are still present, are you guys having trouble transitioning, or are you guys not present and going to lose points for the day? Obviously, TAs, disregard that. How's it going, Brianna?
So far, so good.
Good. How was last week's worksheet in immunology? Because I don't know how how three of you guys are doing with the immunology. Was everything okay?
Yeah. In most of the labs, I haven't gone super heavy into lecture, just kind of more gone through questions and stuff like that. But for this one, I did do the lecture because I thought it was kind of, because there's so much information and it's pretty new to everybody. I did the full lecture.
All right.
And I think that was helpful.
Good.
Because I know I had to go back through the PowerPoint and kind of really hammer down what stuff was because it's newer to me. So that was definitely helpful, and they did well on the worksheet. So
Good.
I think they understood okay.
All right. That's good to know. Hopefully that game was helpful to them to understand some of those concepts.
Yeah.
Give them 3 more minutes. Another 20 more seconds before everybody's actually back in the room, and then we'll begin. All right, hopefully that concept was pretty easy to understand. Let's see how you guys actually did. Did you guys get these particular answers for the respective terms? Right. Majority of you guys have answered. So the majority of you guys are doing well. You guys are getting it. There are a couple of people that want me to explain, so I'm going to quickly do that as this question is relatively easy. Let me share my screen. All right, so going back to the term sensitivity, the sensitivity is for trying to test if a person's actually infected. And so when it comes to testing or seeing how sensitive a test is, the term that's going to be most associated with it is true positive results. So in this particular case, for the true positive results of this particular diagnostic test is going to be 98%. That means that 2% of these people that are actually infected are going to end up getting false negative results. So they are infected, but the results is not, it's 2% not sensitive enough to register that it's positive. So it's going to generate these false negative results. Whereas specificity is trying to see how specific is the test. Is it going to be able to not detect people that aren't actually infected? And so if you're truly not infected and the test has a specificity of 95%, that means 95% of the time that a person isn't actually infected, the results are going to generate 95% of the results as true negative. Now, because we have this margin of error by 5%, that means people that aren't infected but do this test, that means 5% of them are going to end up testing positive. But again, because they're not actually infected, this is known as false positive. So this should have been 5%. All right, yeah, question?
Oh yeah, I just wanted to clarify then. So then the false negatives are actually the complement to the true positives, right?
Absolutely, yes.
Okay, yeah. And then the false positives are the complements to the true negatives. Okay,
exactly. So yeah, so if you can use the color coding that I have here to help generate more sense with that. So if you can associate the sensitivity with true positive and false negative results, then you'll be able to basically tease out what the answer is. And same thing with specificity. If you associate specificity with true negative and false positive, you'll be able to tease out what the actual answer is. All right. So good, you guys are doing well on that. You guys will get questions on those. They're very straightforward, easy points. So if you're still having trouble understanding, either find me after class or ask one of your peers. So for COVID testing, there's basically three main kinds of tests that are out there on the market. And it's all dependent on if you're showing symptoms versus if you want to see if you've previously been infected. And so for people that want to know if they're currently infected, the two tests that doctors and lab technicians will do is do this reverse transcription Qpcr, quantitative polymerase chain reaction test, or they'll do an antigen test. Whereas if you've previously been infected, these two tests do not detect that because they're looking specifically at the virus. Whereas with the previously infected test, we're looking at antibodies, IgM and IgG, which I went over last week, to look specifically at your immune response that has been generated against the virus, assuming that you've been infected. And so we're going to go through each one of these. And again, what I really want you to take away from this is, what is the starting material? What is the assay or the test trying to test and look for? And what is some of the basic science on how this works? So we'll start off with Rtq Pcr, which stands for this whole long name, but you're going to be hearing me say Rtq Pcr, just Qpcr. So for the steps of testing for the virus using Rtq Pcr, you're basically going to take a sample from the patient. You're going to then isolate the nucleic acids from that sample, and then you're going to perform the actual test, and then you're going to analyze the results. I got a quick question. Will taking the COVID vaccine register as an infectious previous infection? Good question. Not depend on the test. So if for these particular tests where it's looking specifically at the virus, no, it will not. Now, because the whole point of a vaccine, which we'll talk more about in tomorrow's lecture, is to generate immunity against the virus, one of the ways you develop immunity, as I talked about in the adaptive immunity lecture, is developing these antibodies that are against the virus. And so the vaccine is basically going to simulate that, except it's not going to make you as sick or even sick at all, depending on your sensitivity to your immune response to antigen. You're going to be you will end up testing positive with this particular, the antibody test, because you have antibodies against the virus. All right, so with a Qpcr, basically the starting material is going to be a nasal swab or trying to get some of the saliva from the patient. So again, remember, the virus infects the respiratory system of people. And so there's going to be productive virus all throughout, mainly the lungs. But as you're coughing up and stuff, there's going to be virus all throughout, like the esophagus and mouth and nose area. And so what technicians will do is end up swabbing these regions to see if there's any virus present. And so what the point of this test is to look at the genetic material. So only the genes of the particular virus, not the protein, the genes. So be very conscious of what it's being tested for. So all this is looking at the genetic material of the virus. And so if the genetic material of the virus is found present from the test, then that means you have ongoing infection, assuming that it's a true positive result. Now, with a negative result, if it's a true negative, then that means you don't have any virus in your body. However, sometimes the test can register as negative for a couple of different reasons. One might have to do with you just recently got infected and there's not enough productive virus that's present. So if virus hasn't been put into the environment, it's still replicating within your cells, then this nasal swab isn't going to get any of that virus. And then the other common thing that people end up testing negative for is because they didn't actually insert the nasal swab far enough to actually collect the sample. And I have to emphasize that using the Qpcr test, it does not tell you if you've previously been infected. It only tests ongoing infection. So be very clear about this. A lot of the questions I'm going to end up asking you relevant to this lecture are going to be questions that you should be able to apply in your own life and understand why these assays work the way that they do. And hopefully you can be a source of knowledge for some of your family members that are interested on how some of these tests work, because it's a common thing that's being done. So you should have a general understanding of how these tests are functioning. So, the technician collects the sample, and then what's going to end up happening in the lab is they're going to end up isolating the nucleic acid. And there's this whole long process that basically purifies and takes out just the DNA and the RNA of a sample and gets rid of any cell debris or organelles and whatnot. And so once you have the nucleic acids, then you can go ahead and actually perform the test. And so when you perform the test, you'll get the samples or the results within hours if you're doing it in a hospital, or as some of you guys have may have done at testing centers, will take a couple of days because they have to get sent all the way to a lab, and the lab has to process all the samples, and then they have to update all the information, versus if you're already in the hospital and you're a patient that's being specifically seen, then they'll contact you immediately. In terms of specialized equipment to do this assay or test, and I'm going to use assay and test interchangeably, but they basically mean the same thing. It's relatively cheap to do, but the most expensive part is the equipment that's used to perform this assay. And the name of this machine is called a thermal cycler. So basically, you're going to mix a couple of things into these little tubes, and then you're going to put it into this machine, and then there's going to be a couple of different heating cycles that are going to occur, which we'll get into in a couple of slides. But this machine can be very pricey. But once you have the machine, the actual cost of running these samples to test are relatively cheap. When performing this particular assay, there's three main components that occur. So there's the reverse transcription aspect, there's the polymerase chain reaction component, and then there's the quantitative component. And so just have a general understanding of the different parts of this, as you might see some of this in the news, and being familiar with this terminology is just going to give you more power so you can actually explain this to people, instead of just looking things up on Wikipedia and not having any idea what it means and extrapolating stuff. So we'll start with the reverse transcription process. So again, the whole point of this particular assay is to test for viral genetic material. And so all genetic material are genes. So not proteins, genes. So if we go back to, first week of stuff, just, there's going to be a lot of review and application of first week material with understanding some of these assays. So remember, the coronavirus has an RNA genome. And as I've stressed in previous lectures, RNA is very unstable. And so in order to actually run this assay, you need to make the RNA stable, and you do this by making complementary DNA or cDNA. And the way that this is performed is by using this enzyme, which I've talked about in the past, reverse transcriptase. And so if you remember when I've talked about in the past, reverse transcriptase is just the opposite of transcription. It's reverse transcription. Instead of making DNA to RNA, you're taking an RNA template and converting that to DNA. And so once you make the DNA is very stable. It's very heat resistant and just doesn't degrade very easily. And so here we have our positive RNA sense genome of the coronavirus. And then you go ahead and add this enzyme, reverse transcriptase, to your mix. And then basically there's some heating cycles that you have to do that basically will end up synthesizing this new complementary strand of DNA. And so once you have this cDNA, then you can go ahead and go to the next step. And I think there's some questions. Is it possible that during the process of prepping the tubes for the machine that there could be systematic error when giving inaccurate results? It is possible. It's more of if you didn't set the cycles properly, which I'll get into in a second. It's more of contamination that is going to end up causing problems. This, and I'll show a slide at the very end when comparing specificity and sensitivity of these three tests. The PCR is the most sensitive of the three tests. And so you usually don't get a whole lot of errors with this one. Usually it's due to user error or some kind of contamination, or not setting this up properly. So once you have what is it? Sorry, I got thrown off a little bit. Okay, yeah. Once you have your cDNA, now you can actually perform the polymerase chain reaction part of this assay. And so what you need to do is actually amplify a specific gene that is relevant to the virus. And so going back to the virology lectures talking about COVID, most of these PCR tests are going to be focused on amplifying genes that are specific to the spike, envelope, membrane, or nucleocapsid. And so, those genes are very specific to the virus. We don't express any of these genes. So even though there might be some human DNA that's present, the PCR isn't going to amplify any of that because we're looking specifically at the viral components. And so within this polymerase chain reaction, there's going to be three heating steps that occur. There's the denaturing, the annealing, and elongation. And so if we go back to DNA replication, a lot of the principles that are applied to this amplification are also the ones taken from DNA replication. And so scientists usually will, study a lot of organisms and how things work, and then we end up using some of this information for our own personal gains to be able to create tests as such. Somebody asked, so the PCR machine basically recognizes self from non-self? Not that. So self and non-self is more of a term when it comes to our immune response recognizing a pathogen. So this is going to be, so what you're asking is more relevant to the antibody test, which I'll expand more. What this test is looking for is if COVID is present within the sample, then you can amplify a specific gene for that particular viral component. So again, any of these. And so if the viral component's present, then you're going to end up testing positive. Hopefully that made sense. And if not, maybe after I get through the whole lecture, then if you still have questions, go ahead and ask me again. I think once we distinguish these three different tests from each other, then it might make more sense. So the first step of any PCR reaction is to denature the DNA. And so a typical temperature that is used for denaturing DNA is 95 degrees Celsius. And so what this temperature is going to do is basically separate the two strands from one another. And the whole point of separating these strands from one another is so you can go ahead and add a primer to your particular sample. So during the step, what's going to end up happening is the thermal cycle is going to end up lowering the temperature to about 60 degrees Celsius. And this is a typical temperature that primers will start to bind to the separated strands of the DNA. And so just like if we go back to DNA replication, a polymerase needs a starting point in order to add on the nucleotides or to synthesize the new strand. And so what scientists do is basically add in primers to the tube that are specific for their gene of interest. So these primers will be specific to one of these particular gene sequences. And so once you have your particular DNA primer bound to your respective strands, then this will act as that main starting point for the DNA polymerase. And again, polymerase is just an enzyme that's going to add nucleotides to a respective strand. And so after the primer binds and the polymerase, the DNA polymerase has a starting point, the temperature is going to be increased to about 62 degrees Celsius. And this is going to be the optimal temperature for DNA polymerase to function. So each of these particular temperatures has a function. So, understand that basic biological principles as there is a reason why there's this cycling that occurs. Once the DNA polymerase binds to the primers, then elongation can begin. And again, the whole point of DNA polymerase is to add DNA nucleotides to the respective strands so it can synthesize a new strand. And so that's basically one cycle. And so with the polymerase chain reaction, you basically have this cycle going on 30 to 50 times. It really depends on your protocol of interest. But the whole point of this is to amplify that specific gene of interest. And so you can imagine by the end of 50 rounds of replication or amplification, you're going to have a lot of copies of that particular gene. And so, we started out in cycle 1 with just 2 strands. And then in cycle 2, we double that to 4 strands. And then here I have an example of cycle 3 where that doubled to 8 strands, and then so on and so forth. And so this is the, the basic summary of what happens during PCR. There's the heating, and then the primers bind, and then there's the amplification, elongation, and this process just continues until whatever you set the thermocycler to. But usually it's around 30 to 50, and that's just going to be enough rounds of elongation to be able to actually visualize your gene of interest. So here we have now the quantitative part of this assay. So within the actual tube that has all of these things, the DNA polymerase, your genetic material, free random nucleotides and whatnot, there's also a fluorescent probe or marker that's within your tube. And the whole point of this marker is every time there's a new round of replication that occurs, there's going to be a light reaction that happens. And so basically the computer has a specific threshold before it starts to register things as positive or if things are there. And so at this beginning part of this curve, we see like there's replication that's occurring, but there's not enough light for the computer to actually detect the actual gene of interest. And it's not until enough DNA has replicated that there's enough light that's emitted that the computer can actually detect, and then you start to see this exponential increase of the amount of replication that's going on. And then basically once enough light has been created, it's just going to be complete exponential, and you're going to have lots of light and that's being detected, and that is going to correlate with the amount of in this case virus or genetic material that's present. And so usually what will end up happening is at the end of the sigmoidal curve, it's going to end up plateauing. And that's because there's a finite amount of reagents or components that are used to actually do this test. And so once you run out of components to basically perform the replication process or elongation process, then it can't do it anymore. So it just kind of just stabilizes and plateaus. And so one great thing that you can do with this particular assay is you can quantify the amount of virus that's present. And so the more virus that's present, the closer it's going to be on the x-axis. So if you started with a lot of virus that's present, then the sigmoidal curve is going to come up somewhere over here, as opposed to a patient sample that has very little virus, it's going to come up much later. And so these right here on the bottom denote the amount of times that the DNA has undergone elongation and replication, that whole PCR reaction. So here is what the results look like if you're actually positive. So again, the sigmoidal curve is going to be generated. Here's where, there's a threshold that the computer needs to register for enough light to be emitted. Once enough light has been emitted, then you can start to actually visualize the amount of replication that's occurring, and then, the plateau process because everything ran out. So this is typically what a technician or whoever's running the sample will end up seeing. If they end up seeing this curve, then they're going to register the sample as positive. Whereas if there is no coronavirus present, then there's nothing that's going to end up getting amplified. And if nothing gets amplified, then it's going to basically stay below this threshold level, and you're never going to end up getting the sigmoidal curve. So, understand the differences between these two and the basic biological principles to do this assay. So let's change gears a little bit to the antigen test. So this distinguishes from the antibody test. Antigen, antibody, there's some similarities between them, but antigen is different. So it's similar to a Qpcr in the sense that the starting material is the same. So you need a nasal swab or saliva from the patient. But instead of trying to amplify genetic material, what this test is looking for is viral proteins or viral components in the patient. And this, again, is specific to looking at if you have current ongoing infection. And so this particular antigen test will look at the basically the main structural proteins of the virus, the spike, the membrane, the envelope, and the nucleocapsid. And so if it's a true positive test, then that means you have ongoing or active infection. And just like with the Qpcr, if it's a true negative, then the test isn't going to detect any virus. But it is possible that if there's not enough virus present or you didn't collect enough sample, then you're also going to get that negative results. And just like with the Qpcr test, it does not tell you if you've previously been infected. And so this antigen test works similar to how a pregnancy test works. There's a test strip that basically has a couple of parts to it that you should be familiar with. You have a control part and a test part. And basically these sections are going to be filled with capture antibody for whatever they're trying to capture. And so in the context of COVID, you basically are going to have antibodies for a positive control test. And so what this is going to do is to tell you that the actual strip worked properly and you can test the results, or whatever the results are, they're actually accurate and reliable. If nothing ends up showing up here, then you can't trust the results. And so when you end up running the patient sample on the strip, you basically, it's going to end up going to this direction. And then, so you have the patient sample in green and the controlled detection antibodies to register that the strip actually worked correctly in red. And so when you run through this strip, if we remember by antibodies are going to bind to specific things. And so when the control detection antibodies make it to the control section of the test, basically you're going to have some binding that occurs and then you're going to see a band that's present. Whereas if you actually have COVID present, then the antibodies in this section are going to recognize specific viral components and are going to end up binding to that region and then testing as positive. Now, if you run a patient sample that doesn't have any COVID present, you should only get one band for the positive control. And because there is no virus present, then you shouldn't see anything in the test section of the strip. And so here you really want to understand what these test results look like and how to interpret them. Because I may give you one of these test strips and I want you to be able to understand how to read them, because it's very important to do this, as you may actually end up running this test on yourself or a loved one, and you want to be able to understand what this actually means. And they're quite simple. So I just explained the basic, biology behind it, but to actually read this, what you really need to first look at is to make sure that the control band came up. So here we have C for control and T for test. And so if the control band comes up, then that means that there's the test is reliable. You can trust whatever the results tell you. Now, if there is no control band that comes up, then the test is going to be invalid because something basically went wrong in the whole process of running the sample through this strip. So if the control band comes up and a test band comes up, then you can assume that you have COVID. And this is assuming that there's true positivity that's present. Now, if a control band comes up, but there is no test band comes up, then that means you do not have COVID, assuming again that it's true negative. And then just to emphasize, if bands or no bands come up on the test, but there is no control, the tests are invalid. You can't interpret anything from them. So now let's talk about the antibody test, which is a little bit more, I would say, complicated. So let's just go ahead and review some basic antibody structures that I've referred to in the adaptive immunity lecture, because antibodies are so important when it comes to a lot of these tests, as we just saw with the previous test. So again, with the antibody, there's two main components that are important for when it comes to fighting against the virus. You have the variable region that's going to actually bind to the virus, and then you're going to have the constant region that is going to be uptaken by innate immune cells via their FC receptors. And so this is to help, phagocytose anything that's bound to the antibody. And so again, with antibodies, they are very specific to what they can bind. They can basically bind to most things. So unlike T cells, which they can only bind to a peptide sequence or a protein sequence, antibodies can not only bind to that, but they can also bind to sugar residues and nucleic acids. And so when we're trying to look for antibodies for this test, your starting material is going to be blood. And so there's two different components of blood that you can use for this test. And so here we have our starting material, whole blood that's just taken from a patient. And then what this blood's going to be treated with is anticoagulants. So anticoagulants basically cause all your blood to, blood factors or clotting factors are going to cause all your blood to stick together and, get very thick. So anticoagulants are going to prevent that process from happening, so we can keep everything in a nice solution. And so what's going to end up happening is you're going to have this blood treated with the anticoagulant, and then you're going to layer it onto this low viscous solution. And so the whole point of this low viscous solution is so after you go ahead and spin or centrifuge, you can go ahead and separate the different components of the blood. And so when you do this particular process where the anticoagulants are present and you spin down, there's going to be three main components or three different layers of the blood. You're going to have your red blood cell layer. So this is where all your red blood cells are going to be, and which makes up about 45% of your total blood composition. You're going to have this they call it a buffy coat, and this is where all your immune cells and platelets are gonna be. And then the most important layer that for this test that technicians need is the plasma. And so this is the majority of what your blood is made out of. And so within the plasma, you're gonna have water, you're gonna have some carrier proteins that move hormones around, minerals like salts and or sodium and chloride. You're going to have some clotting factors that are going to be inhibited because of this anticoagulation. And then you're going to have your antibodies present. So remember, your antibodies are just a B cell receptor that has been secreted into the blood. And so all of these antibodies basically circulate in the blood so it can bind to, in this case, coronavirus. If you don't use any kind of anticoagulant, the blood's going to end up coagulating on its own. And so when you do that, basically after you spin it really fast, then you're left with two components instead of three. And so within these components, you basically are going to be left with a blood clot. And so this is where your clotting factors, your red blood cells and white blood cells are all going to be kind of stuck together. And then the component that you really care about is going to be the serum. And the serum has all of the same stuff that plasma does, but it doesn't have any of the clotting factors as the clotting factors we use to make this blood clot. But just to emphasize, the plasma and serum is where your antibodies are going to be after the blood is processed. The technician is going to take a sample of either the plasma or the serum and test for your antibodies. And so once you have the plasma or serum, then you can actually perform the test. And so the test is looking specifically for IgM or IgG antibodies. And these are going to be specific against the virus. And so a positive test is going to indicate that you've made antibodies against the virus. And so that means that you either have ongoing infection if you have IgM present, or you have had the infection before, but you've cleared it, and that's if you have IgG antibodies. If your test comes back negative, then that means you've never been exposed to the virus. And kind of just going back to one of the questions earlier, if you gotten the vaccine, you're going to end up developing these antibodies against the viral components that are made from the vaccine. So if you're vaccinated, you can potentially have a positive test. The results for this typically takes a few hours because it's kind of a long process when it comes to incubating or allowing some of the interactions to occur. And depending on how the sample is being processed, you may need something called a spectrophotometer, and that's what this is here. And this is usually basically to measure the amount of antibodies that are present within the serum or plasma. If you're just trying to get a positive or negative result, then you don't need this because there's just going to be a color reaction that happens. But if you're actually trying to measure the amount of antibody that's present, then you use a spectrophotometer. And this is, of course, I'm not going to test you on this. Is just a basic, understanding of how this works. There's some light source that shines a specific wavelength onto the sample. The sample ends up absorbing a fraction of that light. And then there's going to be a number that's generated, and that's going to be the absorbance value. And then depending on if you had something called a standard curve, where you have a known concentration of whatever you're looking for, in this case, antibody, then you can back calculate to see how much is present. But I'm not going to quiz you on any of that stuff. That's just for your knowledge. And if you want to know more, feel free to find me after. The cost of this can be pretty pricey. I think I mentioned in one of the other lectures, a lot of insurances do not cover this. And so just trying to send your blood sample out to see if you have IgG or IgM present can range from hundreds to a couple thousands of dollars. So a lot of people don't end up doing this test. And so kind of going back to this timeline that I talked about last week, IgM is going to be the first antibody that's made against a pathogen. And so here we can see this graph that I showed again from last week, that once you have the initial infection, you're going to start to show symptoms, and then the immune response is going to start to kick in. And so the IgM is going to be the first line of defense when it comes to the adaptive immune system. And then as the B cells become more educated, then they undergo again this process called class switching, where they switch their IgM into IgG. And this IgG, it's going to be more specific for the pathogen, and it's going to end up helping the innate immune system recognize the pathogen and kill it more efficiently. And so if you remember from last week, IgM has this pentamer structure. There's 5 of them that are stuck together. And that's because it has low affinity or less attraction to the antigen. So there needs to be more of them in order to actually recognize the pathogen or for it to bind to it well. If your test comes back positive for IgM, that means you have recent or ongoing infection of the virus. Whereas IgG, again, it's more specific for your particular, whatever it's trying to fight against, in this case, coronavirus. IgG is going to last in the blood for months to years, as opposed to IgM, which is only a short amount of time, as you can see by this curve. IgG has a high affinity or a high attraction for the antigen, so it only needs 1 of them instead of 5. And so if you have IgG positivity in your sample, then that means you've already cleared the virus and you've had previous infection. If you are test positive for both, then basically you're kind of in this in between where your adaptive immune system has really taken over at this point because you have IgG present, but you're still in the process of clearing it because you have some IgM left over. But really understand what the purpose of each one of these is. Somebody asked me a question. Are the antibodies we develop with the vaccine the same as the ones we develop without, but stronger? You know, that's an interesting question. So yes and no. So it's specific in the sense that, say, if we end up getting infected with the virus, there are multiple structural proteins, right? There's the spike, there's the membrane, there's the envelope, and the nucleocapsid. Most of our immune response when we're fighting against the virus will develop antibodies against all of those different parts. Whereas, so you're going to end up, having antibodies for all of those different kinds of proteins. Now, with the vaccine's specific to only the spike proteins. If you look at all of the types of antibodies that the immune or the adaptive immune response makes against the virus, the majority of them are against the spike proteins. So with the vaccine, basically the vaccine encodes for like 20 different parts of the spike protein. And so you're really only going to have immunity against the spike proteins of the virus, but because the majority of our immune system recognizes the spike anyways during an actual infection, there isn't really going to be like a main difference in terms of the immune response, except for your body's not actually fighting against the virus. It's just, trying to clear or get educated on specific sequences. So once you actually encounter the virus, then you can go ahead and clear it a lot faster. So here's how this antibody test works. So again, it's pretty long. And so, it's usually a matter of hours before you can actually get the results. And so what researchers will do is make actual like COVID antigen. So, pieces of the viral protein, and then they're going to put it onto these things called plates. And so each plate has individual wells, so you can run multiple samples. And so the antigen is going to stick to the plate. And what we remember is antibodies recognize the antigen. And so we're testing to see if any antibodies within the serum or plasma recognize the COVID antigen. And so again, the healthcare workers will end up taking the blood sample, processing it, so you can either have serum or plasma, and then that serum or plasma is going to be added into the respective plate. And then it's going to, they usually allow it to sit for like an hour to two to allow that antibody antigen interaction. And then after that comes in, you're basically going to come in with another kind of antibody called a detection antibody. And that detection antibody is going to recognize your IgM or your IgG. And so the main point of these detection antibodies is so you can go ahead and see some kind of light or color reaction that occurs. And so basically you have, the antigen, and then you have your IgM or IgG, assuming you've been infected before, bind to the antigen itself. And then you have a detection antibody that comes in. And then with that detection antibody, here we have there's going to be some kind of molecule attached to it. And a lot of the time, this molecule will end up changing to a blue color after you add specific, they're called, substrates, that basically will end up breaking this off and creating some kind of light or color reaction. And so if there's a color reaction that occurs, then that means that there's positivity for your particular antibodies, and that means you previously had COVID before. So here I have a comparison of the sensitivity and specificity of these three tests. So with the Rtq Pcr test, again, it recognizes the viral components of the virus. And this is the most sensitive of all three of the tests. And then it has a relatively high specificity, but in my opinion, I think sensitivity is more important than specificity. because you want to make sure that that you're registering people that are truly infected as infected. The antigen test, which is the one that's least used, and you may have heard it in the news or maybe not. Usually the Qpcr and antibody tests are the ones that most people will talk to or actually use. With the antigen test, it has a very low sensitivity. It has approximately 80%. So meaning that 20% of people that are infected aren't being registered as infected. But it has a high specificity, meaning that if a person isn't infected, then most of the time it's going to register that the person doesn't actually have it. With the antibody test, it's kind of a happy medium between the two, where you have a sensitivity about 90% and a specificity of 97. So really, understand these terms. And if you were to get, again, any of these pieces of information, know how to apply these terms to the different kinds of, true positive versus false positive, et cetera, et cetera. With that, I will take any questions. Actually, I think someone asked me a question. So once the S proteins are identified, hold on. So once the S protein, I wouldn't say that much of a threat. The whole virus by itself is a threat. Yeah, it's just, this is what we recognize because that's what the virus uses to get into the cell. If you guys have any questions, let me know. If not, I'm going to go ahead and add the sign-in sheet into the chat. You guys, again, will have 10 minutes to do this. And yep, let me know if you have any questions.